Hi everyone, I'm the plant propagator and welcome to my channel. Today it is an early morning here in Southwest Florida, so not up to 90 degrees yet, but you can tell it's, well, I watched the weather, it's going to be a warm day today. What I wanted to do today is share something with you about orchid seed development, and I wanted to do this using, explain something to you using corn, maize. And I was at the grocery store. It is sweet corn season here, and I thought, well, this might be a good example, a good way to share some information with you that'll hopefully have a little bit more meaning to you if I use corn as a model or as a way to explain things. So I'm doing this because I like corn. In my previous life with the university, I worked with corn, not sweet corn, but field corn. And there are some similarities, or there's some ways to get some information across about, orchid seed formation and pollination and all those types of things using corn. Corn is a monocot, so it is a plant that has parallel venation, and similar to orchids, which also are monocots, and that's pretty much where the similarities break down. So it is a certain type of monocot. Orchids, so this is an orchid, this is an orchid, there are other types of monocots, but they're in the same general class of plants. Orchids and corn are in the same general class of plants, and that is they're monocotyledonary plants. And you think of this and you look at this and you're going, well, they don't really look like corn, but this one right here does a little bit, doesn't it? This actually looks like corn, and this is a catasetum. There are really a lot of dissimilarities, but from the leaves you may be able to see the parallel venation in the leaves that makes this characteristic of a monocot. That's again where I think the similarities break down a little bit. What I want to do is talk with you a little bit about seed formation, and just so you know, give you a little bit of my background. I have worked for most of my career, and what I'm known for is embryogenesis in plants. And I worked on embryogenesis of maize and other monocots. I worked on rice and wheat also, and some other ones. But embryogenesis means the word breaks down for embryo, which is embryo, and genesis, which is creation or formation of. And so I've worked on this area for a number of years. I'm familiar with it, and I hope to use some of what I know about it to explain to you seed formation in maize and in orchids. All right, so let's take a look at this. I've shared with this some of this information with you before, broken down and separated, but I want to kind of bring it together for you today. So as far as let's take a look at maize, and this is what's interesting. Some of you may be familiar with corn, and this is an ear of corn that I'm going to have tonight with dinner. What comes out of the end of the ear, as you can see here, are these silks, all right? And the silks shown here are this is the stigmatic surface of the corn during the pollination process. And what that means is these are receptors. These are the female receptors for the pollen. In corn, the flowers are male and female, all right? Unlike in orchids where you have both parts of you have male and female parts on the same flower. In maize you have separate parts. So the tassels that come out of the top of the corn plant, that's the male part, and that's where the anthers are, and that's where the pollen comes from. The ears that come out of the side of the plant, this is the female part of the plant. And the stigmatic surface where the pollen lands is on the female part, which is the stigma of this plant. I hope I'm not again getting too complicated, but essentially the bottom line is the silks are the surface where the pollen lands on, and then the pollen grows down into each of the seeds that are on the ear, and you get pollination or fertilization. All right, and that's what's interesting. So in an ear of corn shown here, each one of these seeds that contains an embryo has a silk associated with it. On this ear of corn, there are about 800 to 1,000 seeds on this ear of corn. Each one of these seeds has a silk associated with it, a stigma associated with it, and then each one of the seeds that are in there is pollinated by the pollen that is located on the tassel. And I hope this isn't getting too complicated, but this is how things work, and it's interesting. So what I want you to remember and I want you to understand is that in corn you've got, about 800 embryos that are forming in here. Each of the embryo is in its own seed, and each seed has a stigma associated, which is the silks that grow out of the end of the ear that the pollen then lands on. All right, I'm going to get back to this in a little bit. In orchids, it's a little bit different. And I should mention, I'm not sure whether I mentioned it before, that in corn, when you have a separation of male and female flowers, and there's a few plants that do this, this is called monoecious. And again, I don't want to get caught up in definitions, and this isn't going to be on the test, but that's just what it's called. In orchids, you have male and female parts on the same flower. And the pollen, the structure of the flower is a little bit different. Well, it's very different from what it is with maize or corn. In the orchid flower you have, and I should also say that the monocots, this group of plants, is the most evolutionarily advanced group of plants. So they've developed some interesting features over time. All right, let's get back to orchids. So these flowers have both male and female parts. The interesting thing about orchids is that they form pollinia. So rather than in corn you have pollen that is kind of a wind pollinator, spread by the wind, in orchids you have pollinia, which are sacs of large numbers of pollen grains that are formed within the flower. Each pollinium that's in here, again, it's this sac, contains about anywhere from 500,000 to a few million pollen grains. Orchids form seed capsules, and that's shown right here when you have a successful pollination. And this is a decent sized seed capsule on, I think this plant is this is Dick Smith, and so I did a pollination on this not long ago, about a month and a half ago, and so this is a seed capsule, and the seed capsule is going to get larger. In this seed capsule there's probably about 1 million seed forming. So there's 1 million seed in here. So what that means is that, and what happens, it's the same type of thing. The pollinium that I harvested from one flower was then placed on the stigma, which is similar to the silks here, placed on the stigma, and those pollinia germinated. It's the same process in seeds. The pollinia, the pollen germinated and then went down and fertilized each egg. And so if there's 1 million egg in here, and the pollinia is fertilizing each egg to form the seed, then there's going to be, yes, 1 million pollen grains in each one of these pollinia. And so here's an image that shows a sac, a couple of different sacs of pollinia, and these structures again enclose about 1 million to more pollinia, or pollen. So what that means is then during the pollination process, the pollen germinates, and each one sends a pollen tube down to fertilize an egg. So what's going on in this little tiny flower is that you have 1 million pollen grains that are germinating. So when you look at this flower, you've got a lot going on during the pollination process. All right, so again, the stigmatic surface is the receptor for the pollinia. The pollen germinate and go down and fertilize each. So you have 1 million, probably 2 million pollen grains in the pollinium, and each one of those pollen grains goes down and fertilizes an egg. So each seed in here that is a result of that egg fertilization is a unique individual. All right, they're all different. So you've got 1 million coming from the male, from the pollinium. You've got 1 million immature seed that are in here, and each one of those is derived from an individual pollination. Okay, what's the other thing that we can learn? And so that the numbers are what I want to share with you. It's really interesting because there are so many seeds produced by orchids just because they have, because of something we'll get to in a second. There are so many seeds produced in each orchid that you have to understand that results from 1 million eggs and 1 million pollen that then fertilize and give rise to the fertilized egg, which is the embryo, which then develop over the course of 6 months, in this case of this plant, to give you fertilized and more mature seed. I should say that it's 6 months, but it's anywhere really from 6 months to 9 months to, in some cases of some orchids, 1.5 years. All right, so this, I want to give you, again, that's the idea of the overall process. What makes the seed from orchids different from these seeds? Well, the unique thing about orchids, and the reason that the seeds are small and there's so many of them produced, is if you imagine seeds that were this large, that were produced by this plant, they would be huge. This thing would be the size of this whole table. All right, the seed of maize there's in each one of these seeds there's an embryo, but also in each one of these seeds there's endosperm. So that is the material that is in the seed that feeds the embryo once the seed germinates, once the embryo within the seed germinates. All right, orchids don't have endosperm, so the majority of stuff, and again, it's what makes the sweet corn sweet. It's what makes many of the seeds that we consume good for us, is they contain endosperm that contains, really nice amounts of protein and starch and lipids and all those things that are helpful for the plant when it germinates. They're also good for us when we consume them. All right, the orchids do not have endosperm. The seeds are tiny. The embryos within the seeds are also tiny, but there's no endosperm that can feed the embryo in the seed. There's no endosperm in that seed, and that's what helps to make this plant what it is, and that's what helps to keep the seed small so that the seeds then can be wind dispersed and end up in the right location. All right, so again, it's the endosperm, which if you open up, if you're eating corn next time and you're eating the seed, and you can notice what's in there. So mostly it's just there's the seed coat, and the endosperm is everything inside of that. So it's starchy, but it's kind of half liquid. But then there's the embryo that is at the bottom. So the embryo is at the basal side of each one of these seeds, and you can see it if you look for it. It's a small structure. It'll be white to maybe cream colored, but each one of those in this ear of corn, 800 of them, and there'll be each one of these seed where you're eating the endosperm mostly also has the embryo inside. So next time you're eating this, you can take a look at it. All right, so that's all I have for you today. I wanted to share with you some of the similarities and differences, and I wanted to use between maize and orchids, and I kind of wanted to use this again. It's a little bit of an unusual video today, but it's kind of what I did. I just thought of, okay, this would be an interesting comparison to explain pollination and seed development in orchids versus maize. Similar, but oh so different. All right, that's all I have for today. I hope you enjoyed my video. If you did and you want to keep on seeing them, it would help me out if you can click on like, share, and subscribe to my channel if you're not already a subscriber. All right, that's all I have again, and happy propagating.